Hello, and thank you for joining us for Chardan's Virtual Ophthalmology Summit, where the focus in the current installment will be on the rare ophthalmic diseases. My name is Daniel Gataolen. I'm one of the senior biotech analysts here at Chardan, and it is my pleasure to introduce Lance Baldo, the CEO of Beacon Therapeutics. Lance, welcome. Thank you for joining us.
No, thank you for having me. Excited to have the conversation today, Daniil.
Course, yeah. So the session will follow a fireside chat format, and likewise, I'm excited to talk about the progress that you guys made at Beacon. So to start things off, maybe if you can spend a few minutes to introduce yourself and provide a brief overview of Beacon Therapeutics and the current programs.
Sure. Maybe very quickly to make this part of it a little bit easier, I will go ahead and show a slide here so that we could have a really rich discussion. But my name is Lance Baldo. I'm a physician scientist by training. I've been the CEO of Beacon for about a year now. Most of my career has been in pharma, big and small, including running ophthalmology business at in Genentech here in the Bay Area, where I reside for a couple of years in the past. And pretty excited about the work that we're doing here at Beacon. We have a competitive pipeline of both rare, and we're going to spend some time talking about Larazova today, as well as prevalent blinding diseases of the eye. The most advanced program is Larazova, which is for a condition called XLRP, a devastating disease with no available treatments, which we announced really in the last week, I guess. I guess it's, yeah, just this week that we completed enrollment in the pivotal VISTA study. So we're quite excited about that. We feel very fortunate to be able to relay that we're well capitalized and backed by a global syndicate of blue chip investors that really bring a lot of not just capital, but also expertise to the table, having worked in either ophthalmology, retina, or gene therapy, or some combination of two or three. And likewise, we're bringing together a group of individuals that have expertise in retinal diseases and gene therapy across different functions, whether it's CMC or regulatory, or most recently, as you and I were discussing, Daniil, just before the start of this call, we brought in Dr. Dan Chung, who's now going to be leading our medical organization as our Chief Medical Officer. We have offices in the UK as well as in Boston and another in Florida as well. A little bit of a legacy there in terms of some of the innovation has derived from Oxford and from the UK. But Larazova, which was formerly known as AGTC 501, that was when the Series A investor, Syncona, it was a take private of AGTC, and then eventually really focusing all of the resources on what that group of investors and the initial management thought was the most promising asset, which was formerly AGTC 501, which is now Larazova for XLRP. And so, yeah, just pretty excited to be working in this space right now and for being one of the companies that has a clinical stage asset that's going to be nearing a readout sometime next year. And we're really excited to work with the regulators to bring this medicine to patients.
Excellent. Thank you, Lance. That was a great overview. And with that, let's dive in and talk about your lead program. So Larazova for XLRP. Before we jump into Larazova itself, can you talk a little bit about XLRP? What is this indication? What is the unmet need? I mentioned no available treatments. So what is the current standard of care? And among the RP patients, what fraction do you expect to be able to capture with Larazova?
Yeah, and I do have, fortunately, a couple of slides to help foster this discussion as well. So, for those of you that aren't familiar with X-linked retinitis pigmentosa, XLRP, it falls into the family of the so-called retinitis pigmentosa, which are actually really quite common when you look at across a global population. About 2 million people across the world are suffering from some form of retinitis pigmentosa. One of the most common forms of retinitis pigmentosa are called the XLRPs, and of those, RPGR XLRP is one of the most common. And because they're X-linked, they affect primarily, but not exclusively, male. And of the XLRPs, this is about somewhere between 10 to 20% of those overall RP cases. So we like to say that although it's a rare disease, it's not an ultra rare disease, and it's one of the most common forms of the retinitis pigmentosa. And I wish I actually had better news, but unfortunately, there's no current treatment that's available for these patients. In fact, typically when you talk to clinicians and they're seeing these patients and they characterize them as having XLRP, really everything is just supportive and helping them get their lives in order for the eventual fact that they're going to slowly and progressively go blind. And the way the disease starts, Daniil, is that early in childhood, you have some changes in peripheral vision, some trouble seeing at night, and you might start bumping into objects. By the time you're mid-20s, it's no longer safe to drive. Probably even at that point, somewhat difficult to hold down a steady job, difficulty reading, and really bumping into objects. And by the time you're in your mid 40s, about half of the time you're legally blind with no ability to read and really a lot of difficulty even navigating obstacles and trying to move around your environment as well. So a really devastating disease. And right now there's nothing that's available. And luckily there's a lot of support for developing products within this category. There's a really great group of advocates, particularly with the Foundation Fighting Blindness, and they spent a lot of time making sure they're advocating for science, for patients, for clinicians, so that companies like ours can continue to innovate and try to get these medicines to patients in need.
Got it. Thank you. Let's talk about Larazoa. Can you give us what this construct is, the vector used, any information you can share about the construct, and particular what differentiates it from other gene therapies
Yes.
for therapy? Yeah.
Certainly, certainly. So, this is an AAV2 vector gene therapy that's delivered, as you can see on the left part of this slide. It is delivered subretinally. So, a so-called bleb, about 200 microliters, is placed in the region of the macula. And then that's going to target the photoreceptor cells in that area using a proprietary capsid that has high transduction of both rods and cones. And the promoter is a fairly commonly used promoter within the setting called GRK1, which exclusively targets rods and cones. And then really importantly, and this is one of the differentiated aspects of Larazova, particularly compared to another product called Bodavec, is that it expresses the full length RPGR protein, including a very difficult region called open reading frame 15. So therefore, we're able to actually express the full length protein, which allows to optimize the restoration of not just the rods in the periphery, but also the cones in the central part of the vision as well, because that ORF15 is involved in something called glutamylation, and that glutamylation tags rods and cones and allows them to basically persist and be more healthy over the lifetime of the patient.
Okay, got it. So that's as you mentioned, is a differentiating aspect of Larazoa, right? That it's a full length versus some of the other constructs that are not.
Mhm.
So how important do you believe is it to have a full length RPGR transcript? Maybe you could cite some of the evidence that it's advantageous. Because from what I understand, there have not been these full length transcripts have not been isolated. But correct me if I'm wrong.
No, 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 you're right. But there have been studies that have been done, including out of Professor McLaren's lab in Oxford, that showed that when there were deletions or trouble within the ORF15 region of the wild type gene, it leads to decreased glutamylation of the cones and therefore worse and more progressive vision loss as well. So we do have natural history that would show that having the entirety of the protein, and particularly a full construct of that ORF15 is quite important in both rod and cone function as well. But I guess, Daniil, probably the most important thing is the clinical data are going to speak, and that's why we're running the definitive study. It's our belief, it's our philosophy that in fact the full length protein is going to lead to and yield better outcomes. But that's why you do the study, and that's why we're running VISTA, a sizable, controlled, blinded study to see if that hypothesis is in fact correct. And we do know that there is a product called Bodavec, which was derived from Mira and then acquired by J&J. And they recently had a study that failed to meet its primary endpoint. But the really comforting thing there was there were strong trends in the secondary endpoints, which happened to be the primary endpoints that we're studying within the VISTA study. But even there, the magnitudes of improvement in terms of number of letters seen on LLVA or even microperimetry are not nearly on the magnitude that we're seeing. Now, you have to be cautious, of course, with intratrial comparisons, but it would kind
Mhm.
of fit the hypothesis that we believe that that full-length protein is important to try to yield the best possible clinical outcome for these patients. Because the reality is, and this may change over time or over decades, these patients are probably going to get treated once and only once, at least initially, with one or the other or one product. And so it's really important in our judgment to be able to give the patients the best possible chance they have to restore and prevent, further loss of rod and cone function as well.
Okay, got it. Makes sense. And I understand that making that construct is not quite as straightforward, right? That's perhaps partially why not all the competitors use full length. Can you talk about some of the technical challenges or technical workarounds that you had to do to overcome the manufacturing challenges to build this and make it stable?
Yeah, yeah. Unfortunately, a lot of that work was done prior to my arriving here, but I've been given a bit of the history as well. And so a process that's I'm sure very familiar to you, Daniil, and probably many of the people in the audience is codon optimization. And so it's very likely, if possible, that some of the reasons that chronologically the AGTC program, which is now the Beacon program, was a bit behind chronologically the Mirror program was because the company at that time decided that they were going to spend the time to codon optimize and try to be able to do the best job possible to be able to yield, transcription of as close to the full length RPGR protein as possible. And that did take many months and many quarters, including testing many different codon optimizations until eventually the company was able to narrow, that selection down to what we now call Larazova today. And it has been shown both, ex vivo as well as in vivo to yield fairly good transcription of the entire of the RPGR protein, including that really critical open reading frame 15 as well, Daniil.
Got it. Excellent. And in terms of manufacturing it at scale, does that present any additional challenges?
It does not. So far, everything that we've seen on the manufacturing side is that the changes in the codon optimizations that were done don't yield any additional challenges in the manufacturing process. Now, that being said, these are gene therapies and they're not simple to manufacture. In fact, we've chosen to divest manufacturing to a company called Ascend. And so we're working with Ascend. In fact, we're co-located with them at an office in Alachua, Florida as well. And part of the rationale there, Daniil, is that we believe that our core competencies aren't necessarily manufacturing, but it's the research and the development and hopefully eventually the commercialization of these therapies as well. So that allowed us to get the manufacturing costs off of our P&L and allows a group of experts that we work with very closely to be able to do that manufacturing. And so that definitely allows us to focus our resources again where we think our scientists and where we think our development professionals can have the most impact for patients. And that's in the research and the development side of things versus the manufacturing side of things as well.
Okay, got it. And since we're talking about manufacturing, maybe I'll ask my CMC related questions right away. So that agreement that you have for manufacturing, so you're obviously mentioned you completed enrollment in the pivotal study. So that means you've had sufficient material for that. Is that the same material that would potentially be used commercially? And are there additional inspections, testing, and any of that sort required by the FDA before the BLA submission?
Yeah, yeah. So the product that's being studied, the product version that's being studied in the later part of Vista is the product that's intended to be registered and then launched commercially as well. And so we will have to manufacture more so-called PPQ batches to be able to register those with the FDA. Now, the nice thing, particularly working in rare diseases, is some or all of those PPQ batches can also be the initial commercial supply as well. And so we're gonna manufacture those PPQ batches. We're gonna register those with the FDA, and those are gonna serve as the initial launch lots that we'll be able to use with particularly in the initial U.S. and the European launch markets, Daniil.
Okay, got it. Okay, so let's talk about the clinical clinical trials, clinical data. So obviously, it's been investigated in several trials already, right? We have previously seen data from Horizon, Skyline. Most recently, you've presented updates on Dawn. And as you mentioned, you just completed the enrollment in Vista. So let's focus on Dawn, which would probably be most relevant with the recent data. Um, can you walk us through the design and what that study is looking at and what is its importance for your pivotal study?
Yeah. We're pretty excited about the data from Dawn, and it's really served a couple really important purposes, including giving us more confidence in the endpoints that we've chosen for the VISTA study, but also giving us a lot of confidence that the dose and doses being studied here are likely to yield the clinical benefit that we're hoping for within the pivotal VISTA study. And so this is an open label phase 2 study, and it's a so-called fellow eye study. And I'm going to take a moment to explain this because I realized recently it's not perhaps as straightforward as most people would appreciate. So everyone in this study has had one eye already treated with a gene therapy. In almost all cases, it was some version of Larazova. In one case, it happened to be a Biogen product. And then they come back sometimes many months or even many years later, and now the fellow eye is being treated. And that's what we're looking at here in terms of the results. So the control arm or the control reference point here is the previously treated eye. And the investigational arm here is the newly dosed eye as well. And we looked at two dose groups here, and this is something, Daniil, that distances this study from some of the earlier Horizon and Skyline work. The low dose here is a new and is a different low dose, and this is the same low dose that's being studied in VISTA. So this is about half. If you look at the viral genomic equivalents here per eye, it's about half the dose. So we got high dose for group one, 12 subjects, and a low dose in group two, which is 3 subjects. Now, it's primarily a phase 2 safety study, but really importantly, we're looking at some of these key markers of visual and function, including microperimetry, but perhaps even more importantly, LLVA. And so what we do is we dose these subjects and then we follow them monthly, 3 month, and then eventually 6, 12, I'm sorry, 6, 9, 12 months, and then all the way out to 5 years. And we look at the improvements that we hope to see within LLVA. And these are some of the early results that just get us so excited and get the clinical community so excited. At baseline, everyone was normalized to 0. We basically said that that was your baseline. So all of these are going to be what's called mean change from baseline. That's the CFE mean change from baseline and LLVA score. And what you see pretty remarkably is that already by month 1, you see about an average, about a mean 8 to 10 letter improvement in so-called low luminance visual acuity. And what LLVA is at a very simple level, you take a standard eye chart or Snell chart or BCVA, and you do it in low light conditions, controlled low light conditions, but low light conditions, and then basically see how well the subject can review those letters within those low light conditions. And then 5 letters is a line. So every 5 letters is a line. So basically 10 letters is that these subjects are getting about 2 lines of benefit on an eye chart. And for those of us, and I'm sure it's all of us on this call that have done an eye chart, getting 2 lines of improvement on an eye chart is pretty significant. We then follow them out, of course, month 3, month 6, month 9. You will notice on the legend here, the numbers do drop off a bit. That's just because patients haven't quite achieved those time points yet. So as the data mature, we're going to get more data and more confidence in the data all the way out there. So early improvements by month 1, and we see persistence all the way out to month 9. And Daniil, one other way to look at this, and then I'll pause for any additional comments or questions on this, is so that was looking at things from mean change from baseline and LLVA score. This is now something called a responder analysis. So what proportion of subjects within the study had either a 2 line, which is a 10 letter, or a 3 line, which is a 15 letter gain in LLV response. And you see a very high proportion of subjects having both 2 and 3 line responses across both the high dose group, which we're looking at here on the left, and the low dose group as well. And on the right, a different way of looking at is, these are the same subjects, but just portrayed a different way. Everyone had a positive improvement in LLVA, and a high proportion had either a 2 line improvement or a 3 line improvement in LLVA. So very good results, not just on mean change from baseline, but also looking at a responder analysis. So in other words, it's not 1 or 2 subjects that are driving all this benefit. Many patients are getting pretty significant benefits to Neil in terms of improvements in LLVA.
Okay, got it. Is it surprising to you the magnitude of the fact that just 1 month?
Yeah,
or
yes, it is. Now, typically what you see is usually by 2 weeks, you actually see a worsening of LLVA, and that's because it's a subretinal injection, and the patients do get corticosteroids, topical, oral, and subtenons. And so you initially see a degradation, a deterioration in LLVA, and we kind of expected that by month 2 or month 3, we'd start to see an improvement. But really, already by month 1, we're seeing that improvement. In fact, one anecdote that I can tell you is that a patient came back for their 1 month visit. This is all, of course, blinded to me and to the company, and told the investigator that for the first time in almost 20 years, they were able to walk into a restaurant and see the name of the restaurant. Now, Danielle, you and I take it for granted when we go out to dinner that we could see the name of the restaurant on the sign next to or above the door. But by 1 month, again, this is an anecdote, but by 1 month, this subject within the study was already able to read the sign of the restaurant to which they're going to eat. Again, you and I take this stuff for granted, but for an individual that's living with XLRP, this is a very, very meaningful improvement in their life as well. And yes, to answer your question again directly is, yes, we're seeing those results already by 1 month, which at least for me was a little bit surprising, a little bit surprising. But it does bode well for the mechanism that the drug's working. It's localizing to to the cells, the photoreceptor cells within that photoreceptor layer, and we're getting the transcription of the protein, and then ultimately the localization of the protein to the appropriate place within those rods and cones to yield. The other thing that was, I guess, maybe not surprising to me, Danielle, when you think of the mechanism, is we're not just stabilizing vision. Many products can stabilize vision. We're actually getting improvements in vision, and my belief in it is on that early in the course of disease, because this RPGR protein is associated with both protein movement within the inner and outer segments of the photoreceptors, but also energy metabolism, you can actually get rods and cones that aren't yet dead. You can get them to improve their function, and therefore actually get these improvements in these measures like LLVA. And then hopefully, and I think that's what we're seeing right now, you had mentioned Skyline and Horizon, then you're getting stability where you're actually hopefully maintaining the function of those rods and cones once you get that initial benefit that you're seeing here in Dawn, Daniil.
got it. Yeah, that makes sense. And Lance, what was the mean time interval between treatment of the first and the contralateral eyes here?
I don't have it memorized, but it's on the order of years. So this has been, and there's quite a range, but I think on median, it was about 2 years between the first and the second eye in this Dawn study. That'll be different than how we intend that this is going to be used clinically. And in fact, we're kicking off a small safety study called Landscape, where we're going to take 10 subjects and we're going to give the dose in one eye and then very shortly after in the other eye, just to make sure it's safe, which we of course believe that it will be because we have numerous years of experience both with drugs like Luxturna as well as with Bodivec as well to show that it's safe to administer one eye and then the other eye. But that's something that we're kicking off as well, a study called Landscape, Daniil.
Okay, got it. And I don't know if you are doing that or if you have enough data to do this analysis. Um, in terms of correlation between the time that has passed between the first and the contralateral eye treatments, if are there any data, or do you expect to collect any data that shows the shorter the time interval, the greater the effect?
No, we don't have enough data to be able to show that. And the other thing that's a little bit confounding, Daniil, is that the initial eye treatment, as I mentioned, was a variety of different product formulations and also doses as well. So it's not a really
Mm,
good apples to apples comparison. The
gotcha.
contralateral eye treatment was all the same procedure. The bleb was placed in the same place, and it was the same product by process as well product. So a lot more similarity in how this eye, for the results you're seeing, was treated.
Hmm. Okay. And I guess another question on this, this being a contralateral eye, which essentially is given, gene therapy, second dose, right, to the same patient. We know that it's been done safely with Luxturna. For other indications, clinical trials show that it's doable without any safety, notable safety effects. Can you comment on any safety signals that you observed in Dawn? Do you have to use a more comprehensive or enhanced prophylactic regimen for contralateral eye?
Yeah, we've had a fairly consistent corticosteroid regimen that we've applied in the later phases of development. In fact, the corticosteroid regimen being used in Dawn is either exactly or largely the same regimen that's being used in Vista right now. And as I mentioned, it is an oral taper, topical with drops, and then a sub-Tenon's injection. And to your point, Daniil, a lot of the, at least the early side effects that you see in the subjects within Dawn, or really in any gene therapy, subretinally delivered gene therapy study, are either usually associated with the steroid regimen itself, so it's serving a necessary purpose, but also has the concomitant side effects that you might see with it, or from the surgery itself, because it is a subretinal surgery. It does require a vitrectomy. You are lifting the retina off the layer of the eye, and so it's not without some risk there as well, Daniil. The good thing is the risk benefit on this. These patients, it's a devastating disease with nothing else that can be done, and so the risk benefit for this particular disease state is extremely good as well. And so far, in the doses studied in the later stages of the clinical development program, we haven't seen any AEs or SAEs that were associated with the actual gene therapy itself. With the surgery, yes, and with the corticosteroid regimen, yes, but even those have been quite manageable. And again, when you put in the context of the risk benefit for this disease, it's actually been quite impressive, in my opinion, Daniil.
Okay, got it. So you've had an opportunity to collect quite a bit of patient data over the years, and share those with KOLs and retina specialists. Can you share some of the feedback that you're hearing from advocacy groups, retinal specialists, KOLs, et cetera?
Yeah, no, I could probably spend 20 minutes on this just in itself, but there's a lot of excitement and a lot of enthusiasm. And I know that this is maybe a very indirect measure, but the session at Arvo where we presented the most recent Dawn data, Dr. Panisi presented the most recent Dawn data, was standing room only. There was no room to even sit down if you got to that lecture hall a little bit late at all. And then as soon as he was done with his presentation, a lot of people left the room, which just showed me that a lot of people wanted to be there to kind of see these emerging data as well. And I really wanted to give a shout out to FFB and the Foundation Fighting Blindness. They do an amazing job advocating for their patients, including most recently where they drafted an open letter regarding the fact that they felt that that the Lumio study, which showed the failure on the primary endpoint, but some encouraging trends on the secondary endpoints, they really questioned and wondered whether or not there was something that could be done for patients in terms of trying to get agreement from the agency to bring Bodivec forward. We love this and appreciate this because it just shows that number one, you've got a patient advocacy group that's doing what they should be doing, which is advocating for science, advocating for patients. It also shows that the unmet need is there as well. And all the things that they're talking about, including encouraging signals on measures like microperimetry and LLVA, is exactly what we're seeing within the Dawn study and exactly what we're studying within Vista as well. Yeah, so we're just really pretty excited about this, and hopefully, it's going to be a different world now. The idea of standing on the shoulders of giants. Luxturna and the Spark team really did a lot to kind of create awareness about these therapies, including the importance of genetic testing, which is still not done across the board, but it's done a lot more routinely now than it was pre Luxturna as well. And so we do believe that when we finally move through the clinical development, through the registration process, and to the commercialization, that there's going to be fewer obstacles that Spark had to kind of overcome when they launched the first ever gene therapy for the back of the eye, Daniil.
Got it. Yeah, that's great. To see the patient and advocate support. I did hear about that open letter too, and it seems the talk in the community about the primary or the more important or relevant endpoints being LLVA, which is the primary endpoint in your study. With that, let's shift gears and talk about the phase 3 VISTA trial. If you can, maybe spend a minute or two to walk us through design, how many patients you've enrolled, and maybe more importantly, what are some of the key differences or key learnings from your prior trials that you've implemented into VISTA?
Yeah, I mean, there's many in terms of kind of the key learnings that we've implemented here, not the least of which is the dose selection. The lower dose that was studied in the earlier Skyline study was an order of magnitude lower than the high dose studied here, and it showed no treatment effect. So the team made what I think was a really good data-informed decision that that was not a good dose to bring forward in phase 3, and so we knew that a dose about half the level of the high dose was in fact going to be safe. And so the decision was made to move forward the 3.7 to times 10 to the 11th as far as the lower dose as well. We also learned the optimal location of the so-called bleb, where's the best place to put the bleb, and improvements in the actual surgical procedure itself. So how can we make sure that we can get the medicine to exactly the right place with the least challenge as possible? And so the company, the team has gotten pretty good as well. And probably the key thing, which you've already alluded to, Danelle, in your questioning, is making sure you have the right endpoints, what I like to call discriminatory endpoints. So, there's great clinically meaningful endpoints, but if it's clinically meaningful but not discriminatory, in other words, if it doesn't tease out the treatment effect, it's not particularly useful. We saw in some of our earlier data that while we were seeing trends in improvement in something called maze or visual mobility, we just didn't think that we were going to be able to show enough treatment effect to justify having that as the primary endpoint. So we do have the maze within the study, both a physical as well as a virtual maze, but we've chosen to look at LLVA, low lumens visual acuity, and mean change from baseline and mean microperimetry as the two primary endpoints within the study itself. And we can get into a little more detail here in terms of exactly, how we're going to frame that within for the U.S. regulators as well as the European regulators. And this is the design of the study. It's three different groups, the high dose group, the low dose group, and then a group of untreated controls. The comparisons here are going to be between each of the doses individually and the untreated control group. The primary endpoint is a 12-month, and we're collecting data, of course, well before that and all the way out to 5 years. But the primary objective, the primary endpoint is going to be 12 months and looking at the changes in visual function for the blinded and masked dose group compared to the untreated control. And there will be the opportunity for the controls for the fellow eye to be eligible after 12 months to be able to move over for ethical reasons as well as for science to move over and actually get dosed in the other eye as well. And I'm super proud to say, Daniil, in fact, the press release went out, it's fully enrolled. We completed enrollment in the last couple of weeks, and just completely thrilled and thankful to the investigators, to the patients, to their families, to the Beacon team for the excellent work that's been done there as well. We are just completing the surgeries because you get randomized, then you actually have to have the medicine implanted, of course. So we are in the process of completing those surgeries and intend to have that all done by the end of this month, Daniil.
Okay, excellent. So with that, just doing a simple math, 12 months primary endpoint. So we should, and some time for analysis, we should see the data in a little over a year from now, right?
That's right. Before the end of next year, figure you got to finish all the surgeries and then you'll do database lock, database cleaning. And so allowing a few months for that before the end of next year, we'll have the top line readout for the VISTA study as well. And really excited to have that. And that's going to be, those are the primary data. They're going to support the BLA and the MAA.
Got it. Makes sense. And Lance, so you alluded to selecting the primary endpoint. LLVA versus Botovac maze was the primary endpoint there. When you were having those discussions with the US and European regulators in terms of the pivotal study design, was there any pushback to include the maze as a primary endpoint, or what can you maybe share how those conversations went in you explaining what you believe would be the most appropriate endpoints for the study?
Yeah. Yeah. Let me see. I'll close this. I don't think I have a slide on that. I'm going to close it right here. But the well, first of all, it was a series of correspondence and communications back and forth with the agencies and particularly on LLVA with the FDA. What the FDA wanted to see on microperimetry was something that we didn't think would be clinically possible, which was defining the loci that you think that you're going to. So when you do microperimetry, right, there's a whole bunch of loci, spots in the back of
Mhm.
the eye, defining the loci that you think are going to have the most meaningful improvement, and then showing that you can get at least 5 decibels improvement there. The problem is, and one of the good things is that we're placing this 200 microliters of medicine in the back of the eye, and we're covering a whole lot of those loci. And some of those loci are getting, pretty profound improvements, but trying to determine exactly which 7 loci are going to have the most profound improvements, we thought was going to be near impossible. And the data were telling us it was going to be pretty close to impossible. And so we realized that microperimetry, at least how the US regulators wanted to see it, was not going to be something that we thought would be feasible or viable. Now, in Europe, the European regulators believe in the clinical meaningfulness of something called mean change from baseline and mean microperimetry. So you look at a whole bunch of points in the back of the eye, and then you look at the mean improvement over time. And if you can show an overall mean improvement, a decibel, decibel and a half, 2 decibels, then you can show that you're doing something clinically meaningful for these subjects. But that was not going to be acceptable to the US regulators. So the engagement that we had there was, well, how about something like LLVA? And the US regulators have told us that they believe that LLVA is a clinically meaningful endpoint in the setting of XLRP. Now, we're very fortunate that we have a number of regulatory designations, including RMAT, and we do have an upcoming FDA interaction where we're going to kind of finalize the agreement with the FDA about how we're going to handle the overall approval package and what's going to exactly be done in terms of statistical analysis and what's going to exactly go into the BLA as well. But the European regulators have expressed their acceptance of mean change from baseline and mean microperimetry as a clinically meaningful endpoint, and the US regulators have expressed their willingness to accept LLVA as a clinically meaningful endpoint in this disease state. Now, we're getting a little bit into the weeds right now, but we still haven't locked the statistical analysis plan, and so it's possible that we may choose to do something. Do we do LLVA for both regions with microperimetry and support? We're going to be engaging with the regulators to make some final decisions on that as well. But one awesome thing about the way the team drafted the protocol is that the protocol gives us that optionality because we're capturing both of those measures in a very rigorous way to be able to leverage them in the statistical analysis approach.
Okay, got it. Makes sense. And with respect to those interactions, particularly with the US regulators, given fairly recent changes at the FDA, have you noticed any changes in your interactions?
No, I consider ourselves to be very fortunate. And Daniil, I often like to tell the team, like the FDA is often considered to be like, it's the monolith. It's this giant, bureaucratic organization. The reality is it's a number of, often very committed, hardworking individuals that really want the best to advance science and to bring innovation to patients. And we've been very fortunate that, at least so far, we've had no turnover in our review team, project manager. And in fact, the engagement with them has really been quite good, where they're responding to our queries. And as I already mentioned, granted our request for an upcoming face-to-face meeting as well, without any pushback whatsoever. I will also say, Daniil, kind of on a kind of holistic level, the words, the rhetoric that we're hearing out of the agency seem to be very supportive about what we're trying to achieve in terms of, making sure that we have the right bar in terms of data for these areas of high unmet need where the diseases themselves are fairly rare, trying to speed or at least trying to find a better glide path to approval as well. Now, the
Mhm.
actions have to meet those words, Daniil. I like the words, and the words are very comforting. But hopefully, we'll learn over the next months and next quarters whether the actions are going to meet those words. But so far, every indication is that it seems to be the case. But time will tell. Time will tell.
Okay, got it. So in the last couple of minutes, a couple of more general questions. So you completed Series B last year, 170 million. Can you tell us what milestones you expect this capital to carry you through? Is that sufficient to complete the VISTA study? Is that sufficient to dig into the rest of your early pipeline? What are the plans there?
Yeah, no, certainly. No, very, very thankful to have raised July last year a large round, $170 million round that brought in Forbion and TCGX into the mix, as well as adding to Oxford, Advent Life Sciences, and Syncona. And so we have a really nice syndicate of investors that, as I mentioned earlier, they don't just bring the capital to the table, which we're very thankful for, but also a lot of knowledge and experience in this area as well. And those monies do take us, the key milestone, which you and I were just discussing a little while ago, they do take us past that pivotal readout, but it's not enough money to take us through to the commercialization or launch or even into a lot of the work that we'll need to do on our second asset, which we didn't have time to talk about today, which is BTX-001, which is being developed for dry AMD as well as an intravitreal injection. So we will have to do something to make sure that we keep the company capitalized, and whether that's a strategic deal, whether that's going to be debt, whether that's going to be another private round, or whether that's going to be potentially listing in the public markets, time will tell. And we're definitely keeping all of our options open in that area, Daniil, as well.
Got it. Okay. And the last question that we like to conclude those meetings with, what do you think investors are still missing about Beacon, either the lead program or early other programs, anything else that you think is being overlooked?
Yeah, I mean, this might be a good slide to chat through that. Well, I think I guess for better or for worse, there's not too many companies that have late stage programs that are fully enrolled. And as you kind of pointed out earlier, it's a matter of measuring on the clock how long until we understand the kind of product that we have and what the path of registration looks like. So very thankful that we completed enrollment in that study. And I think that's really important for investors to understand. And then we're going to get that readout before the end of next year, and that's going to ultimately support registration and then ultimately commercialization. And so I think those are some of the key things. And whereas I think, in fact, I don't think, gene therapy, particularly as of late, there's been some kind of mixed sentiment as well. I personally believe that Beacon is in the perfect place right now, which is within the eye, because you've got a organ that has immune privilege, which is fantastic for studying these gene therapies. We've got a disease that's well characterized in terms of what exactly is causing the underlying disease. And we've got a kind of, a gene augmentation, a protein augmentation approach where it's kind of lock and key. And then we've got the endpoints. That's another great thing about ophthalmology, where you have these meaningful endpoints that can be done within these studies to show that you're actually getting the treatment effect that you're looking for. So I feel like Beacon, we're perfectly set up for success, and I hope we're a really compelling story to whatever the future investors look like, whether that's private company, I'm sorry, public investors or potentially strategic partners as well, Daniil.
Mhm. Yeah, excellent. Well, thanks, Lance. On that note, I think we'll wrap this up. Again, I really appreciate your time, and thank you for participating in our summit. Thank you.
Same here. We really appreciate the invite. Thanks, Daniil.